So our third speaker is Dr. Andreas Tolias. Dr. Tolias is a professor and Brown Foundation Endowed Chair of Neuroscience in the Department of Neuroscience at Baylor College of Medicine. His research combines systems and computational neuroscience with machine learning to uncover network-level principles of intelligence, particularly in perception and decision-making. Today, we'll be hearing about his group's pioneering work creating the MICrONS open data resource. So please join me in welcoming Dr. Tolias.
Thank you. Thanks for the organizers, and thanks for organizing this nice meeting. So I'll start by asking these questions: are these dogs or muffins? This is something that is innate and very easy, I hope, for everybody here in the room. But this is actually a very hard problem in artificial intelligence, and something that is not even with the most modern methods being solved. And I think this quote here by Einstein is sort of indicative of why maybe there is this gap between current AI systems and intelligence. And in the talk today, I will give you sort of our approach in trying to understand what makes brains special and machines unable still to solve this type of problems. And I think at the heart of it is that probably the key ingredient of intelligence is the ability to generalize outside your training distribution. You know, an organism or an agent that has never seen the zoomed-in versions of either dogs or muffins cannot recognize them, an AI system. And it has to be trained in this domain, whereas, you know, humans and animals are really good at this. And we would argue that, you know, there's different levels of generalization, and most of the sort of current AI systems, and this is an ongoing debate, but even like ChatGPT, there is evidence that sort of mostly extrapolating or maybe interpolating within its training distribution. Whereas, you know, we humans, if you see, for example, to a child, the first time you see a pink elephant, you can recognize it, so you don't have that type of problem. So what makes it special? How do we go about understanding how brains can do this and machines cannot? And we would argue that a key ingredient in this is understanding representations. And of course, this goes back to what Mackenzie was talking about and the David Marr analogy, understanding how is information encoded or represented in the cortex. And for example, here you can see a scheme, a very cartoon scheme, where you live, let's say, in a world where there's shapes and colors. And if you encode them along this sort of factorized code, where you have neurons that have pure selectivity to either color or shape, the first time you will see a square, in this case a blue square, you'll recognize it because you can just learn to project in these, you know, subspaces or neural dimensions, and then you'll be able to do inference. Or even you have a mixed selectivity code, if it's, you know, tilted in a particular way, you can get this type of disentanglement. So of course, understanding representations has a very, you know, long history in neuroscience, and some of the, you know, here is a famous discovery by Hubel and Wiesel done at Johns Hopkins many years ago. And as many of you may be aware of, is that when they were trying to do these experiments, they were using spots of lights because they were students of Kuffler, and Kuffler studied the retina. And it was by accident that one day they had this dirt. You can sort of see it here. Actually, this is a 60-minute interview of David Hubel explaining this discovery, that they found serendipitously that neurons are tuned to edges, and it opened up the whole field of visual neuroscience, essentially. And if you look at a lot of these discoveries, and you actually look at the history, and you even talk to the people that did them, like Charlie Gross. I think he actually did this discovery here at MIT before he moved on to Princeton, was by accident. It was serendipitous, or people having very strong intuitions of what they should be studying. And this is, I think, true about place fields, right, Matt? And grid cells for sure. You know, in fact, you know, it was like, I think they even had the data, and some student or postdoc went and like squinted it and said, yeah, it looks like grid cells. So if you look at the cortex, let's say, of the macaque, there's dozens of visual areas. And if you look at, you know, there's like, now we have our work and others, and a lot of work by the Allen Institute, you know, we find that maybe hundreds of different cell types, interneurons. So if we're gonna like make discoveries at this rate, it's gonna become, you know, very, very difficult in our lifetime or in many lifetimes to understand how to really understand these computations and how to really understand, for example, right now, you know, I see familiar faces or new people that I haven't met, but I met maybe during this morning or whatever. How is it that our brain really represents this information and understand it in a more complete way, and hopefully even come up with a normative understanding? So why is it so difficult in vision? I mean, I'm sure maybe you've seen different versions of this, but if you have a monitor and you have 256 by 256 pixels and you just turn them on and off, you get 2 to the 65,000 possible patterns, and there's only 2 to the 300 electrodes in the whole universe. So you're dealing with a very large, even if you constrain yourself in a natural image manifold, it's unfathomably large, and it's not clear how you go about it. If you combine it with behavior, and you go into completely free behaviors, you're just not going to have enough time in many of, just not the lifetime of the animal, but our own lifetime and more, to really understand these systems and the fact that there's so many different areas and cell types and circuits. So today I will describe you one method that we developed in the last couple of years, and there's another 2 pieces of papers, actually, one done here at MIT and one across the river, that are related, but there's some differences, by Jim DiCarlo and Mark at the Livingstone's lab. And we call this method inception loops, that I think provides a path forward in maybe trying to somewhat resolve this sort of problem, or create a systematic methodology to study representations. And first I will describe the method and the original work, and then I'll describe 2. We've done a lot of different experiments in different, in both in mice and macaques. I'll describe 2 discoveries that we recently did, one in monkey and one in mouse, and then sort of wrap up by introducing the MICrONS project. So the first step, inception loops have 4 steps. The first is based on all the wonderful technologies, a lot of it funded by the Brain Initiative. We now, and I think Ali Pasha there yesterday, and also Tim Harris has developed amazing technologies. We can record a lot of data, okay? We can record data, for example, here's an experiment. We can record 5,000 neurons simultaneously, and we can show it 5,000 images in a matter of 2 hours. So we can get to 25 million training points, which we're starting to be in par with large data sets that people use in machine learning to train models. And then again, thanks to the sort of revolution in deep learning, which is mostly a technological revolution of having GPUs and being able to scale up the algorithms to train these models, we can build highly predictive models. We have ground truth data here because we're recording, we're showing in images, we have the responses, we just have to build a data-driven predictive model. And we can do this, and we can predict the neurons very well. And now once we have this model, we basically have a functional digital twin of the system we want to study. And now if you think about it, we can scale this model up, we can replicate it and run it on 1 million GPUs or 1 million supercomputers. And now we can run gazillions and gazillions of experiments to test hypotheses and make discoveries, okay? We can also do some things that are smarter than, for example, since this is a differentiable model, we can use back propagation to ask Hubel and Wiesel's question, what are the optimal stimuli to drive these neurons, or what Charlie Gross did in inferior cortex by discovering facets. And when we first did this, and here you see an animation of this, it's actually a real optimization in the model for one specific neuron that sort of rediscovers the orientation selectivity of a Gabor function. But when we looked at it in mouse visual cortex, this is the first time we did it, and this was a few years ago, and at the beginning we didn't even believe it for actually a year or 2, and we were not even planning to test it because we thought the model was extrapolating outside its training distribution. We found that in the mouse, unlike what we read in textbooks, most of these neurons did not look like Gabor filters, but they have more complex patterns, like, for example, edges. And if you searched in natural images, in thousands or hundreds of thousands sometimes, you would find these features in natural images. So here is like an edge, here is something that looks like a checkerboard, here is like a crescent, like a very close crescent, here is something that looks like a T-junction. So now, this is all done in silico. Of course, you have to verify it in vivo, and since these are chronic recordings, now we can go back, show the stimuli, and do a lot of controls, and you can read about it. And now the fact that you get this very strong diagonal metrics means that the stimuli that we synthesize for each neuron were optimally driving the responses compared to thousands of other stimuli that we tested and many other controls. when we did this in the monkey, just to see, okay, is it like completely wrong, this idea of V1 being Gabor filters and edge detectors? That was not quite true. We're still studying the monkey further, but it's obvious that in the macaque we got many more Gabor-like filters, although even in monkeys there are other tuning functions that violate the Gabor. So then we extended this in area V4. Now area V4 is an area in the monkey where it's a higher visual area in the ventral stream, thought to be important for pattern recognition. It's been studied extensively, and in fact there's some very nice work done here at MIT by Jim DiCarlo's lab, finding optimal stimuli for multi-unit activity using a Utah array. But we wanted to study the topological organization of V4 using depth probes. This was before, we started this before the beautiful NeuroPixels came about. Now we're doing it with NeuroPixels, but we use NeuroNexus probes. And we spent a lot of effort doing very well, developing algorithms to isolate the neurons as well as we could and quantify the isolation. And we wanted to look at the organization of this right now of these neurons, of the activity of these neurons. And when we optimize these neurons, as you can see here, we found quite interesting complex patterns. A lot of them look like sort of textures. Some had like these eye-like features, like higher level features, and some had like things that look like sort of more shape-selective neurons. And now we're also doing this in the color domain. So then when we looked at them, we wanted to see if there is an organization into this system. Are these sort of MEIs, the most exciting images, are they organized in any way? And to do this, we used a contrastive loss very similar to what Mackenzie talked about, but in this case, it's just what people use in computer vision, where we use data augmentation to build a representational similarity space, a low-dimensional representational similarity space that we can bet all the MEIs. And the way I want to think about it, you take the MEI of a cyclical neuron, now you augment it, you rotate it, you scale it, you sort of introduce augmentations or image manipulations that you would classify them as being part of the same image. So those, you keep them to be close to each other in this embedding space, and different neurons are further away. And when you do this, you get this sort of clustering. Now you use a different set of data, and what we saw is that these neurons now in this embedding space form these functional groups that if you looked at the MEIs of different neurons, they looked very, very similar, or they were rotation or sometimes scale versions of each other. So they had this sort of transformations that were interpretable, because that's how we learned the space. But then you could see that different neurons that had different MEIs clustered together, and this space wasn't like homogeneously, covering the space. And the advantage of doing this so one question is, what's the relationship between these types, these functional groups, and all the stuff that is known about transcriptomic cell types or morphological cell types, or even neurons in different layers? And this is an ongoing project that we're doing with Tony Zaidor and Jay Gremier. Now, the other advantage of doing depth recordings, and we're very excited, as I said, to use the NeuroPixels now, is that you can look at this old idea of columns. Now, columns is something that was sort of initially discovered by Mountcastle, and then Hubel and Wiesel found the orientation columns, but it's a very controversial thing where cortical columns are an organizing principle of the neocortex, at least of the primate neocortex. It's a very attractive idea because it would break the vast complexity of the neocortex into sort of more manageable modules that then we can focus and understand, and then start interactions of the modules. So it gives us a sort of, if you want, circuit level scale to understand computation. And when we looked at these most exciting images in this embedding space, they would not just form clusters, but the MEIs of individual neurons would also form columns. So now it begs the question, like if you were to use these more nonlinear system identification methods in different parts of the brain, systematically analyze it, are we going to find that cortical columns are a functional organization of the cortex? And of course, the final step is that you have to verify this in vivo. It's much more challenging to do it in the monkey. These are non-chronic recordings, so everything has to happen within a session. And we spend a lot of effort sort of accelerating the closed loop, the inception loop pipeline, so it can be run within a time frame of about 2.5 hours. And then that's where we get single cell stability, single neuron stability to be able to run these experiments. It's much easier if you have to do chronic recordings and get the same neurons, like, for example, with imaging, like some of the stuff that Ali Pasha has shown and what we've done also with 2-photon. And once we run this very fast in closed loop, you can see that, the MEIs are, the most, they're dry. I mean, we verified that basically these most exciting inputs are driving the neurons optimally. Now, this is just the tip of the iceberg. The MEI is just like the tip of an iceberg, and it's not even clear. I mean, it's for sure not the only, it's just going to be a very narrow way of trying to understand the representation. A very important thing in understanding representation is, for example, to study this at the population level, and this is something that we're doing. We're doing inception of the whole neural manifold, and also to look at individually how these manifolds are composed by looking at invariances, and we have a paper on that for those. Actually, I'll skip this and get back to it later. Something on invariances where you study the whole population manifold, or you can look at brain states. One is on bioRxiv, the other one is published, and you can see how brain state or internal brain states modulate the tunic function in the color domain in mice. And this is a very nice, interesting study done by Katrin Franke in my lab, where she showed that when mice are in special light conditions during dusk and dawn, when they are out and they're being hunted, their selectivity changes towards UV light to be able to detect predators. So this was all discovered through this inception loop. But now in the last remaining few minutes, 4 minutes, I want to show you another study that we recently did. It's also on bioRxiv, looking at contextual modulations. So if you, as I showed you earlier, like, with this example of the muffin and the dog, how you interpret things depends on context. In this case, what is the black spot there, whether it's an eye or a raisin, depends on the context. And of course, this is a very fundamental principle in neuroscience, where we know that neurons, even in the retina, and this is throughout both at the cognitive level, perceptual level, but also the neural level, they're not just modulated by what we call their classical receptive field, but they're modulated by the surround. And the dogma, and this is, there's decades of research done this mostly in the monkey, where it's been shown that if you have a preferred feature in the center and it's covered by the same type of features in the surround, you get a very strong suppression. You get a much stronger suppression if you have these homogeneous or congruent stimuli, and less of a suppression if they're incongruent. Now, we can run this experiment. This was done by Jacun Fu and Katrin Franke in the lab, where they collected data under these conditions where we build models now that these neurons, these models were very good at predicting center-surround interactions. Then we computed the MEIs like before, but now we condition on the stimulus, and now we can generate a surround response that either further activates the neurons, and this has to do with the amount of contrast we're putting, or suppresses it. And to our surprise, what we found this pattern completion, that an exciter is around, completed the pattern, and an inhibitor around disrupted the pattern. And as you can see here in a few examples, I'll just show one here. Unfortunately, I cannot point, but if you see, let's say, neuron number 4, it looks like an oriented Gabor, and the excitatory pattern completes the Gabor where the inhibitory pattern disrupts it. And this is not consistent with, the monkey literature. We're trying to do these experiments in monkey now. It's not consistent with a lot of ideas about predictive coding or redundancy reduction. And we've done some modeling work to show that this is more consistent with a hierarchical probabilistic model where Bayesian inference is implemented and the responses are modulated by an image prior that has to do with natural image statistics at the larger scale. Okay. And here is, again, what the model is predicting, and this is the verifying at the bottom in vivo that indeed this type of stimuli excite and suppress. So to summarize this part of the talk, inception loops are a very powerful method. They can be applied across many different species to study study representations or optimize a dynamic system. We're doing this also, as I'll show you in a little bit, with dynamics now. So, and I've shown you that in mouse, we find something that was unexpected. In macaque, we find these functional cell types that may be related to transcriptomic cell types, an ongoing study. And also we find these cortical columns that may turn out to be fundamental, but it remains to be seen. And then we find this pattern completion. Now for the remaining two minutes, I will give you an analogy of what do we mean by if we let's understand all these representations, and we can algorithmically understand the brain. In theory, we're able to even build a brain that does exactly what, let's say, our brain does or an animal's brain does. But that does constitute understanding. So I'll give you an analogy with a toaster here. Let's say you have to figure out how a toaster works, and you've never seen a toaster and you're an alien. There is sort of two opposing traditionally views how to study this, and you can see brain science, like going all the way to anatomy and physiology, being split in this way. One is a more structural approach, and this is also my lab is doing a lot of that, where you study cell types, circuits, anatomy. And the other one is like a functional approach, which is what I did when I was a graduate student here at MIT. They were not paying us a lot back then. And, we would use an iron board to also make toast. And frankly speaking, it works very well, and it does the same job. And that's kind of the physiologist approach. You just want to understand the algorithm and the representation. And that's fine, and you maybe can build smarter AVI, but if the toaster breaks down, you won't be able to fix it. And since NIH is the main funding agency that pays us, and also we want to study diseases, we need to bring the two together. And to do this, the early pioneers understood this. Of course, if you look at these Barlow diagrams or Hubel and Wiesel, they're all about cell types, wiring diagrams, and function. But that's something that is kind of missing. It's a very difficult thing to do. There's a lot of experiments done, but it's a very difficult thing to do. And a few years ago, so what we want to bring is to bring all these modalities together. And a couple of years ago, we were very lucky to be given this opportunity through IARPA. It's a funding agency like DARPA, but through the intelligence agency. And it was a collaboration between Baylor College of Medicine, the Allen Institute, and Princeton to do this at the time, and I still think it's a very heroic experiment, where we imaged the activity of 100,000 neurons across 4 visual areas in one mouse, shipped the mouse to the Allen Institute, and they did high-resolution electron microscopy on this same data. And this data is publicly available. It has actually 200,000 neurons in the volume. There's glial cells, other types of cells, 100,000, about 100,000, I think it's 75,000 actually imaged with very good signal to noise. And maybe Dimitri presented some of the metrics yesterday. And also there's 523 million synapses, and all these data are publicly available. And we're also using them now to try and understand mechanistically some of these principles that I've shown you earlier. To make this easier for the community, Eric Wang in my lab built what we call towards a foundation model of the brain, where we collected many mice, hundreds of thousands of neurons from many visual areas of the mouse. And we built a foundation model, a core model that was trained on all this data, and then it was, through transfer learning, similar to what was shown in the previous talk, feed it to the readout of this mouse, of this MICrONS mouse, and we are hoping to have this on an API very soon, where we can run in silico experiments. And the nice thing about this model, we have already verified it, that it wasn't just predicting. This was all trained on movies, on natural movies, so it's a dynamic model. It wasn't trained on static images, but it not only predicted very well to the dynamic images, but also to different image domains. And with this, I would like to acknowledge the people in my lab that did the work, in particular Edgar Walker, who is at the University of Washington now, who was the main figure in developing the exceptional in my lab. Now he's a professor in Seattle. Fabian Sint, Catherine Franke, that did a lot of the work, and Jakun Fu on the central surround, and Konstantin Willike and Kelly Restivo that did the work on before. And of course, the MICrONS team that collected this data. Thanks.